Hi, my name is Arianna and I belong to the SwitchHD, a research project funded by the European Marie Curie Action. I am a fellow in this project working at the University of Tübingen.
Today I will present you my research plans in SwitchHD rats, a model of Huntington disease. Huntington disease is a progressive fatal autosomal dominant disorder comprehensively described by George Huntington in 1872. The cause of the disease was discovered much later and consists of an expanded CHG repeat, codifying for glutamine, located in exon 1 of the Huntington gene. While in normal conditions the HTT gene carries a maximum of 35 repeats, a repeat expansion with more than 36 CAGs in the Huntington gene can cause the disease. Longer repeats anticipate the age at onset, which on average is about 40 years. The HD mutation causes neuronal dysfunction and death. From a morphological point of view, a main characteristic of Huntington disease is an early and marked degeneration of mediospinal neurons in the striatum. However, other neuronal populations are affected as well, and multiple brain areas show prominent atrophy. This can be observed, for example, in MRI scans, like in this picture, showing the brains of an HD subject on the left and a normal subject on the right. Patients with Huntington disease experience devastating symptoms. They lose their motor abilities and develop cognitive deficits and neuropsychiatric disturbances. Currently, there is no disease-modifying treatment for HD. In order to develop successful therapies in humans, preclinical studies and animal models are necessary. SwitchHD rats are a recent model of Huntington disease, carrying the full-length human HTT gene and all regulatory elements. A first characterization of this model shows that SwitchHD rats reproduce several aspects of human HD. Part of my plans focuses on the characterization of this model for neuropsychiatric-like behavior. Neuropsychiatric disturbances are common among patients with HD and HD gene carriers and can precede the onset of motor symptoms. Frequently reported are depression, anxiety, irritability, and impulsivity. But important symptoms include also deficits in social cognition and emotional recognition, as well as altered sexual behavior. SwitchHD rats have been shown to display changes in anxious behavior. We will then characterize them for depressive-like behavior, and to do that, we will look at behaviors in rodents believed to be representative of depression in humans. We will therefore access anhedonic-like behavior with two-bottle preference test and behavioral despair by using forced swim and tail suspension tests. Because relevant in Huntington disease, we will characterize also social behavior in SwitchHD rats. In particular, we will examine primary forms of social behavior in rodents, including social interaction and recognition of conspecifics in the three-chamber social test, as well as sexual behavior and functionality during a mating test. A second part of my research aims to investigate epigenetic features in the SwitchHD rat model. The term epigenetics refers to changes in gene function that are mitotically and/or meiotically heritable and that do not involve changes in DNA sequence. Epigenetic mechanisms are sensitive to the environment and may play an important role in modulating the effects of the environment on gene expression. epigenetic alterations have been associated to DHD pathology. Indeed, changes in multiple epigenetic markers have been reported in various HD models and tissue from HD patients. By taking a step forward from the basic characterization, we aim to investigate the interaction between environment and genotype. To this end, we will manipulate the rearing environment of BAC-HD rats in order to create a positive environmental condition obtained with social and environmental enrichment, and a negative condition achieved by applying stress. We will then measure the effect of these treatments on DNA methylation, which is an epigenetic marker, and on gene expression. More details on these experiments will follow later. This is all for today. Thank you for your attention.